It's for me a pleasure to introduce today Dr. Albert Giralt. Albert has a degree in psychology from the University of Barcelona. Then he got his PhD in neuroscience at the supervision of Jordi Albert, studying the role of BDNF in the context of Huntington disease. He then moved to the lab of Jean-Antoine Giraud in Paris, for 4 years to deepen on the molecular changes during synaptic plasticity processes. In 2018, he came back to Spain to the Institute for Neuroscience at the University of Barcelona, where he establishes his own group. His current main line of research are focused on the hippocampal synaptic plasticity in health and disease in the context of several neurological disorders. And today's presentation has the title of Astrotherapeutics in Neurodegenerative Disorders and the Role of BDNF. Albert, thank you very much for being here, for your willingness to share with us your work. And the floor is yours, so please go ahead. You can upload your presentation and remember to put it in full screen.
Yeah. Do you hear me, first of all?
Yes, I hear you.
And now, do you see my screen?
Yes, we do. And
okay.
Now we see your presentation, yes.
Okay, thank you very much. Thank you very much, Miguel, for this nice introduction. And thank you to all the audience to stay here today in front of the screen to hear my talk about my research. Yes, as Miguel said, my name is Albert Giralt, and I am a Ramon y Cajal Principal Investigator in the Medical School of the University of Barcelona, and I'm affiliated in the Department of Biomedicine in the same university, in the same medical school. As he said, Miguel, I started my own research, or my team, let's say, in 4 years ago, in 2018. And the main focus of our research is about hippocampal function in health and disease. And today I'm going to talk about one of the sub-aims of this main line of research, which is about astrotherapeutics in neurological disorders. So what I'm going to do is to talk about astrocytes from a gene therapy and cell therapy perspective, and how to manipulate them genetically in order to be therapeutical tools in the context of several neurodegenerative diseases. As I guess many of you already know a lot of things about astrocytes, but anyway, let me introduce you a little bit by doing a brief summary about astrocytes from a classical perspective. As many of you know, astrocytes at the beginning were viewed as the glue of the brain, but fortunately, although these cells were 10 times more in numbers than neurons, but fortunately, the later research discovered many other functions instead of that, involving astrocytes in many functions of the brain, of the brain function. For example, astrocytes in subsequent research were demonstrated to provision of nutrients to the nervous tissue. As you can see here, they regulate. It was shown that they regulate cerebral blood flow, that they have a role in the maintenance of extracellular ion balance, and that they have a role also in the repair and scarring process of the brain and spinal cord following different types of injuries. In the context of neurological disorders, the classical view of the astrocytes was to use them as just a marker of astrolysis and reactivity and neuroinflammation, as you can see here, and immunohistochemistry against a typical marker for astrocytes, the GFAP marker, in a healthy tissue, and in a neurodegenerative context, they increase their morphology, the intensity of GFAP, and the branching. So they were classically used just as a marker, and usually the GFAP levels and the astrolysis correlated with the severity of the histopathological hallmarks of different diseases. Subsequent research was focused in the underlying molecular mechanisms and studying the interaction, in one hand, the interaction between astrocytes and microglia and their role in the regulation of general neuroinflammation. Also, intracellular pathways were discovered, like the role of NF-kappa beta, or in extracellular pathways, like the role of cytokines, interleukins, and chemokines. And the picture was increasing more and more progressively. But the one substantial impasse or a step through was observed or occurred 15 years ago, more or less, in which there was, let's say, a so-called Astrocytes Revolution, let's say in some words. And one of the main hallmarks of this Astrocytes Revolution was the discovery of calcium waves that were observed in this particular cell type. In that moment, it was supposed and hypothesized that calcium waves in astrocytes was a way in which these cells communicate between each other. And also other research teams demonstrated that these calcium waves were used for astrocytes to communicate between astrocytes and neurons and vice versa, and also to synchronize neuronal networks. Other research groups discovered the so-called gliotransmitters, and some of them were produced and released by astrocytes, like, for example, glutamate, glycerine, nitric oxide, prostaglandins, and ATP. This first in-base or first hallmark in the Astrocytes Revolution opened the door to new discoveries, unexpected and unprecedented discoveries, like, for example, the ones that are listed here. Later on, astrocytes were described to transfer mitochondria to neurons. You know that neurons have a high energy demand, and sometimes they have damaged mitochondria, and astrocytes provide with some of them to recover the neuronal function. Also, it was described that astrocytes supply the blocks of neurotransmitters, or another thing that was discovered that was unprecedented was that astrocytes can regulate synaptic plasticity by phagocytosis or other processes. In this context, at that moment, so more or less 10, 15 years ago, one of the main outcomes of these studies resulted in this in the definition of the tripartite synapses. So not only there was a presynaptic and a postsynaptic component in the neural tissue, but also there was a third component, which were the astrocyte processes that surrounded some synapses in order to modulate their function. And how they do that? The astrocytes, for example, some mechanisms are the gliotransmitters like ATP, glycerine, and glutamate. But at that moment, we started to wonder whether a novel and potential gliotransmitter could also play a role. And we talked about BDNF, brain-derived neurotrophic factor. So at that moment, we were in particular super interested with this neurotrophin, but at that moment, it was considered that BDNF was mostly neuronal. So first of all, there were many questions to be answered. Like, for example, do astrocytes produce BDNF, or do astrocytes even though deliver BDNF, in which context, and which was the role of this BDNF? BDNF, as many of you already know, is classically viewed as enriched in synaptic terminals inside vesicles with a proform, and this proform is cleaved in order to produce the mature form. When released upon synaptic activity, BDNF, the mature form can bind to the high-affinity TrkB receptor to induce downstream signaling pathways involved with neuronal differentiation, survival, and synaptic plasticity, involving mechanisms or molecular pathways such as ERK, AKT, and phospholipase C-gamma. On the other hand, when the proform is released and is not cleaved, this proform is also able to bind to specific receptors like P75 receptor to induce opposite phenomena, like, for example, apoptosis, LTD, and growth cone retraction. So this bimodal or this bidirectional role of BDNF at that moment made the emergence of the theory or the hypothesis of the yin and yang proposal of the BDNF function. So this hypothesis or this model states that depending on the molecular context, BDNF can play a beneficial role in neuronal function or a detrimental role in neuronal function. So in this context, it was described also that BDNF was hugely reduced in different diseases, neurodegenerative diseases, like, for example, in Alzheimer's disease and Huntington disease. BDNF was a core protein that was described to be downregulated, and in many, many models, sorry, I will use the pointer, and in many, many models in which BDNF levels were restored, the several parameters and phenotypes in different models of these diseases improved. So it was very promising. But in contrast, in other contexts and other neurological disorders like temporal lobe epilepsy, it was described that an excess of BDNF during the temporal lobe epilepsy progression could be involved with aberrant neuronal activation and consequent excitotoxicity. So the yin and yang theory was emerged, and it had to be taken into account. But let's go back to our initial question. So at that moment, so some years ago, it was there was not too much knowledge about BDNF from astrocytes, and we decided to verify whether BDNF is expressed and delivered by astrocytes. And we did several experiments to answer this question, and I will show you and summarize some of them only as a representative ones. For example, we tested whether BDNF was expressed in astrocytes compared in primary astrocytes compared to immortalized cells like HEC293. And as you can see here, the BDNF levels, the mRNA BDNF levels were much more expressed in astrocytes than in these immortalized cells. This was the first step. And then we did other experiments in which we cultured again primary astrocytes in vitro, and we stimulated them with TNF-alpha, which is a very well-known activator of astrocytes, and we collected the superneuron or media and the cells or pellet lysates to compare the BDNF amount or content extracellular versus intracellular. By performing ELISA assays, we observed that in cell lysates, so intracellular BDNF, was reduced upon TNF-alpha treatment, whereas in the media, upon TNF-alpha, BDNF was increased. This experiment demonstrated that BDNF levels is not only expressed, but also released from astrocytes in physiological conditions. So the next step was to move to another question. And since BDNF has been classically observed as a mediator of synaptic plasticity, we were wondering if BDNF from astrocytes is also playing a role in neuronal plasticity. To answer this question, what we did is this experimental design in which we co-cultured wild-type neurons with wild-type astrocytes. This was the control condition, and we compared these cultures with these cultures in which we co-culture wild-type neurons with BDNF knockout astrocytes. In these conditions, we evaluated the structural synaptic plasticity first by using the Scholl analysis and evaluating branching and dendrites complexity in these two conditions. As you can see here, the branching parameter evaluated was the number of intersections, and this branching was significantly reduced in wild-type neurons co-cultured with knockout BDNF astrocytes. In the same direction, we also evaluated another parameter, which was synaptic or dendritic spine density in the same experiment, well, in a different experiment, but with the same design. And we observed that as the dendritic spine's density was reduced in wild-type neurons co-cultured with knockout for BDNF astrocytes compared to their controls, which were wild-type neurons co-cultured with wild-type astrocytes. So, okay, with all these results, we answered some of the questions. So BDNF was produced, BDNF was released from astrocytes, and BDNF from astrocytes played a physiological role in synaptic plasticity. And then what we did is to move to the next question, and is to use astrocytes from a gene therapy and cell therapy perspective. And we took into account that neuroinflammation is a very common hallmark in all neurodegenerative diseases. And one of the most important markers of this neuroinflammation in terms of astrogliosis is the increase on GFAP expression. So we wanted to take advantage of this and to propose the following model. So what we wanted to do is the following. First, we have that astrocytes. We have shown that astrocytes, in normal conditions, they produce and deliver BDNF in physiological contexts. What we wanted to do is to manipulate astrocytes in order to produce and deliver BDNF only in the required tissue when required. So this means that it should have a spatial and temporal self-control. So the idea is that genetically modified astrocytes, in normal conditions, they produce and release BDNF. But we wanted to do is in the context of neuroinflammation and consequently in the context of neurodegeneration, these modified genetically modified astrocytes could be able to produce and release more BDNF levels in order to counteract the detrimental effects in the context of a neurodegenerative disease. From another perspective, our model also could be summarized in this scheme. Let's imagine a concrete brain region in which there is a zone without apparently neuroinflammation and a zone in the same brain region in which there is astrogliosis or a clear neuroinflammation. From our model, what we wanted to do is to produce and deliver more BDNF only in this brain region or in this affected brain region, but not in this one, because as I told you before, there is a yin and yang effect of BDNF. So an excess of BDNF could be beneficial, but also detrimental depending on the molecular context. So we wanted to avoid that by doing this. So here you have a spatial resolution, so only in the tissue, in the diseased tissue, and a temporal resolution, because only when there is astrogliosis will be BDNF production. So which is the tool to accomplish with this objective? So we generated the main tool, which is this transgenic mouse model. What we did is to generate a transgenic mouse model that overexpressed BDNF under a GFAP promoter. As I told you before, I think GFAP is a typical promoter from astrocytes and correlates with astrogliosis. So higher astrogliosis correlates with higher levels of GFAP. And our intention is that when in the context of astrogliosis, BDNF would be overexpressed or overproduced in astrocytes. Here you can see the construct, and apparently these mice were normal, and also the transgene was expressed only in the neural tissues, but not in other tissues. So the specificity was accomplished, and here we have the body weight, but we performed several analyses, biochemical assays, morphological, histological experiments, and behavioral paradigms, and everything was normal. So in principle, these mice in normal conditions were normal. So after generating this tool, this main tool, we moved to the next step, and we tried to apply this tool to the first neurodegenerative disease. In this case, it was Huntington disease. As many of you already know, Huntington disease is characterized, as you can see here in these slides, is characterized by a severe degeneration of the basal ganglia at the earlier stages, and later on there is a degeneration also in several cortical brain areas. The symptoms that are concomitant with this degeneration are related with motor coordination alterations, but also psychiatric disturbances and cognitive skills deficiencies. The cause of Huntington disease, as many of you know, is an expansion of the CAG repeats in the exon 1 of the mutant huntingtin. And there exist several mouse models to study the disease, and for example, the exon 1 mice, and the knock-in mice, and the full-length mouse models. As you will see in this project, we use the R62 mice. But first of all, what we wanted to do is to accomplish another step, which was to verify whether these astrocytes, these transgenic astrocytes, were optimal in a gene therapy perspective. So if they were transplantable, or if it was possible to graft them, and if they were functional. So to do that, what we did is to use nude immunodepressed mice as a mouse model, and we performed some grafts of these wild-type astrocytes in one hand, or in another cohort of mice, we grafted transgenic astrocytes overexpressing BDNF under the GFAP promoter. And after this grafting, we perform quinolinic acid injections in the middle of these grafts. And as maybe you know, but just to introduce you a bit, the quinolinic acid injection is an acute and toxic mouse model of Huntington disease that recapitulates some, but not all, of the clinical features of Huntington disease, mainly the histopathological features. So what we did is to verify the implementation of these astrocytes after grafting. As you can see here, they survive, and they relatively integrated in the tissue, and they were a bit encapsulated as predicted from other works from other research teams. On the other hand, in terms of functionality, here we can see the effects of a quinolinic acid injection in nude mice graft with wild-type astrocytes, and in nude mice graft with transgenic astrocytes expressing BDNF under the GFAP promoter. As you can see here, I depict the lesion size, which is bigger in the wild-type conditions and smaller in the transgenic conditions. So it seems that these graft astrocytes, transgenic astrocytes, were neuroprotective. We did this experiment in a short time period. 7 days after grafting, we performed the quinolinate acid injection, and it was neuroprotective. This is the volume of the lesion size. And they were also expressing much more BDNF levels in the target tissue compared to wild-type graft astrocytes. And you can see also it's remarkable that wild-type astrocytes also increased. Well, in the conditions, in wild-type conditions, there was also an increase of BDNF, but this increase was much higher in the case of transgenic astrocytes grafted in nude mice. We also repeated this experiment to test for longer times, 30 days of graft, and then we performed again the quinolinic acid injection, and they were still neuroprotective, and they were still expressing more BDNF levels, correlating with this higher neuroprotection. So after verifying that these astrocytes can be grafted and they are functional, then we moved to the transgenic mouse models. As I told you before, we used the R62 mouse model. The R62 mouse model expressed the exon 1 of the human gene with the CAG expansion. And we crossed this R62 mouse model with the GFAP BDNF transgenic mice. We predicted that these double mutant mice would express or would show much better phenotypes compared to R62 mice, so an amelioration of the phenotype. We expected that. And we choose the R62 mouse model because they show an evident and clear GFAP immunoreactivity in the striatum compared to wild type. And as you can see here, we verified this by Western blot. GFAP levels in the striatum were increased in the 2 mouse models expressing the exon 1 of the Huntington, but BDNF levels were decreased only in the R62 alone, and fully, well, no, not fully, but significantly recovered in the double mutant mice. So just to insist a little bit, this increase on BDNF would be explained by this increase of GFAP because we have this transgene, the GFAP BDNF. So the BDNF expressed under the GFAP promoter. Then we moved to the neuropathology and we evaluated the striatal volume in these mice. And as previously described, R62 mice show reduced volume of the striatum that was significantly recovered in the double mutant mice. On the other hand, we also evaluated the density of EM48 positive nuclear inclusions, that is this very important hallmark, neuropathological hallmark in Huntington disease, which is the accumulation of fragments of the mutant Huntington in the nucleus. And this parameter was observed to be reduced in the double mutant mice compared to the R62. Then we further evaluated the phenotype of these mice by performing by testing the motor coordination skills in these 4 groups of mice at different speeds, at 16 RPMs and 24 RPMs. We used the rot-a-rot test, in which we place the mice in a rotating rot and we count the number of falls during the progression of the disease, 9, 10, until 12 weeks of age in the 2 speeds. As you can see here, the R62 mice alone, they showed a higher number of falls compared to the rest of the groups, and in the double mutant mice, this parameter was ameliorated, significantly ameliorated in both speeds. In 24 RPMs, maybe at some point was more clearer. So after these successful results, what we did is to test for synaptic plasticity. As I showed you before, BDNF from astrocytes could play a role in synaptic density, dendritic spine density, and synaptic function. And what we did first is to use fixed brains from the 4 groups, and we stained them with presynaptic markers or postsynaptic markers such as BGLUT and PSE95, respectively. As you can see here in the quantification of the number of positive clusters for each of the markers, in R62, the number of these clusters, synaptic clusters, was reduced in both markers and significantly recovered in the double mutant mice, while in the case of PSE95 was partially recovered. Then we moved to more physiological parameters, and we tested, we performed electrophysiological recordings in acute slices from the 4 groups in collaboration with Eduardo Martin, that at that time he was in Albacete, in the University of Castilla-La Mancha. And we observed that basal synaptic transmission, as you can see here in this graph, was reduced in the RSX2 mouse model and completely recovered in the double mutant mice. And this was indistinguishable compared to the wild type and transgenic mice. So there was a recovery of this basal synaptic transmission. Another remarkable finding was the effects on synaptic fatigue. So we measured synaptic fatigue by using the PS amplitude in the pair pulse facilitation protocols. And as you can see here, from a basal condition, after subsequent stimuli, the presynaptic component becomes more and more reduced in control groups. But in the mice that express the GFAP BDNF transgene, this synaptic fatigue was ameliorated or delayed. So this is also an important finding, we think, because it suggests that BDNF from astrocytes play a role in synaptic plasticity and in synaptic fatigue. So, okay, this was the first part of the objective, to study the role of BDNF from astrocytes in the context of Huntington disease. And then we moved to another disease. In this case, we wanted to determine if this tool could be beneficial in the context of Alzheimer's disease. Alzheimer's disease, as you know, is mainly characterized by a degeneration of the hippocampal formation and also in different cortical brain areas. Concomitant with the neuropathological degeneration, there are several cognitive deficiencies like the loss of memory and spatial memory deficits. And to explain the disease, well, this disease is multifactorial, and there are at least two main theories or paradigms that explain the disease, like, for example, the tauopathy that induced the formation of tau neurofibrillary tangles in one hand, and on the other hand, we have the formation of A-beta plaques that also play a substantial role in the synaptic plasticity deficits in models and in human beings. To study the disease, there exist several mouse models, like, for example, the 5-per-FAT, which is an A-beta amyloid model, the P301S, which is a tauopathy mouse model, or the triple transgenic mouse model, just to cite some of them. There are many, many. In this project, we focus with the 5-per-FAT mouse model, and I will tell you why in the next slides. So it's also remarkable that the topic or the idea that BDNF could be beneficial for Alzheimer's disease is very old. So as far as I know, from 1997, it was proposed that BDNF could be a therapeutical target for Alzheimer's disease. And it's still a hot topic in the field, as you can see in some reviews, proposing BDNF as a therapeutical target. I think there is a microphone switch on. Well, but however, there are many, many caveats and problems to be solved. Well, anyway, in this context, I think that by using the 5-per-FAT model, I can clarify a bit more our idea or our model. As you can see here in this double immunofluorescence against GFAP, the astrocytes marker, and the beta amyloid marker, you can see this is the dentate gyrus, and you can see in yellow the plaques formation, and they are surrounded by super reactive astrocytes. And here in this part, the astrogliosis is much less evident. So from our model, we predict that BDNF will be more expressed and more therapeutical in these regions than in these other regions in which it's not so required, let's say. I'm not saying that there is no disease in this place, but since this is an A-beta amyloid model, it is supposed that the neuropathology, plaques formation, and dystrophic neurites are mostly localized in these regions. So what we did is, again, to use this transgenic mouse model, the 5-per-FAT mouse model, which has two transgenes inserted in the same region. The first transgene is the APP with 3 mutations, and the second transgene is the presenilin-1 with 2 mutations. And the neuropathology of this mouse model is super evident in terms of beta amyloid plaques formation, but also, and importantly for us, in terms of neuroinflammation. As you can see here, GFAP immunoreactivity increases very early in the disease, so as early as 4 months of age. Also microglia, but we are more interested, of course, with astrogliosis. And we did the same design. We crossed the 5x5 with the GFAP BDNF mouse model, and we observed that what we expected is that this, again, as occurred with the Huntington disease mouse model, we hypothesized that these double mutant mice would show much better phenotypes than compared to the 5x5 alone. So what we did is, as we did for the HD mouse model, we validated the mouse model. We observed that GFAP levels were super increased, much more than in R62 mouse model, and that BDNF levels in the hippocampus were reduced in the 5x5, but completely restored in the double mutant mice, as predicted. In this case, we deepen on these biochemical changes, and we evaluated the downstream signaling of the BDNF tract B binding. For example, we evaluated the activation by analyzing the phosphorylation levels of ERK, AKT, and PLC gamma, and we observed that ERK and PLC were reduced. The phosphorylation levels were reduced in the 5x5, as you can see here, and completely recovered in the double mutant mice. Then we evaluated many cognitive parameters from these 4 groups, and we performed the passive avoidance, the novel object location test, and the Y-maze spontaneous alternation. In the first case, just in case, I introduce a bit the test. In the passive avoidance, we place the mice in a chamber with high levels of light, and then the mouse prefers to step through rapidly to the dark chamber, but in this moment, the mouse receives an electroshock. 24 hours later, the mouse is replaced in the same chamber with the high levels of light. But then in this moment, normal mice spend longer to step through to the dark side because they remember in principle that the previous day, they were tortured with an electroshock. As you can see here in the training phase, the time to step through was super short, and in the testing phase was much more longer. But in the case of 5x5 mice, this time to step through was reduced and partially recovered in the double mutant mice. In the case of the novel object location test, we put the mice in an open arena and we present them with 2 similar objects, and 24 hours later, we change the location of one of these objects, and the normal mouse will explore or will show more higher preferences to explore the new located object. As you can see here, then the preference was evident in all the groups. All the group of mice preferred to explore the novel the object placed in a new location, except for the 5x5 alone, which performed in a random fashion. And this performance was significantly recovered in the double mutant mice. And finally, similar results were observed in the Y-maze spontaneous alternation task, in which we put the mice in a Y-maze, in a transparent Y-maze with different objects surrounding the maze in order to create a spatial map. And as you can see here in the board, we counted the number of triads. And the 5x5 mouse model performed in a chance level, whereas the rest of the groups performed higher than chance level. So in general, there was highly recovered of several cognitive skills in these double mutant mice compared to the 5x5 alone. Then we moved to the neuropathology, and we evaluated spine density and morphology in the pyramidal cells or neurons of the cortical layer 5. And also in the CA1, the pyramidal cells of the CA1. As you can see here, we observed a significant reduction on spine density in 5x5 mice that were partially or completely recovered in the double mutant mice. It is interesting to note that when we evaluated the morphology of these spines, we observed a specific affectation of the thin spines in both regions, actually. And this affectation was significantly recovered in the double mutant mice, not in the case of mushroom spines in the cortical layer. So it is important to note that these thin spines have been considered to be the learning spines because they are smaller, plastic, and flexible. In contrast, mushroom spines are more stable and they contain higher amounts of AMPA receptors, NMDA receptors, etc. So it seems consistent that the thin spines are the most affected and that actually the BDNF from astrocytes restore specifically this spine type. Then we moved to the electrophysiology recordings, but this time we did a collaboration. We did it in collaboration with Albert Giralt and José María Delgado from the University of Pablo Olavide in Sevilla. And we perform electrophysiological recordings in anesthetized mice, in the 4 groups of mice, by stimulating the CA3 and recording in the CA1. As you can see here, after, with the experiments, we observed that many parameters were normal in the 4 groups. Basal synaptic transmission and presynaptic function were normal and preserved in these mice, but I want that you focus your attention in the induction of LTP. In this experiment, the team of Albert Giralt et al., they induced LTP in vivo. And as you can see here, there was an enhancement of LTP in the double mutant mice, in the 5-per-fat GFAP-BDNF mice. So this result, I think, is super important and interesting because it indicates that BDNF from astrocytes, again, in the hippocampus, are involved with the generation and enhancement of long-term potentiation, which is a model of cellular synaptic cellular plasticity. Well, so this was the case of Alzheimer's disease, and then we moved to temporal lobe epilepsy. In this case, I say from the beginning that the results were completely the opposite, and I will explain you better in the following slides. So, in the context of temporal lobe epilepsy, it is pretty demonstrated that there is a smaller and even though brighter hippocampal formation, and in the most severe cases, there is also hippocampal sclerosis and degeneration. So epilepsy or temporal lobe epilepsy, the origin or the cause is unknown, but often associated with many environmental and genetic factors, like, for example, mutations in the Relin gene, or brain tumors, or traumatic brain injury, among others, many others. There are also many mouse models, like acute and toxic models, like, for example, the pilocarpine or kainate or other models using drugs. There are also electroshock-induced seizures or audiogenic models. In this context and in our project, we use mostly the pilocarpine and kainate models. And as I told you, the results and the outcomes were completely the opposite. So we performed the pilocarpine model as a temporal lobe epilepsy model in the 2 genotypes, the wild type and the GFAP-BDNF transgenic mice. And as you can see here, after pilocarpine treatment, the appearance of these transgenic mice was much worse than compared to wild type, also treated with pilocarpine. So this is just a picture, but quantifying this by using the Sherpa assessment, we observed that the main score of these mice was much worse in the transgenic GFAP-BDNF mice compared to wild type treated with pilocarpine. Supporting this result, we also evaluated locomotor activity in the open field, and it's highly described or previously described that these models of pilocarpine, they show a hyperlocomotion in the open field, and we validated this in the wild type mice treated with pilocarpine, but the GFAP-BDNF mice, the affectation was so severe that they expressed, instead of hyperlocomotion, they expressed hypolocomotion. Concomitant with this more severe phenotype in the transgenic GFAP-BDNF mice, we observed a massive neuronal death in the hippocampal formation in these transgenic mice compared to wild-type mice, in both cases treated with pilocarpine. And as you can see here, the differences were mostly localized in CA1 and CA3. In line with this, what we observed is that again, we validated the model. GFAP levels were increased in both genotypes upon treatment with pilocarpine. As you can see here, the 2 increases. But mature BDNF levels were increased only in the transgenic mice, but not in the wild-type mice treated with pilocarpine. In contrast, pro-BDNF was increased in both genotypes upon treatment with pilocarpine. So one of the readouts of this result is that the more severe phenotype observed in the GFAP-BDNF levels could be explained by this increase in mature BDNF, but not for this increase on pro-BDNF. And in this work, we also specified, we localized the increase of BDNF in astrocytes, as you can see here, a little bit in wild-type mice treated with pilocarpine, but this increase on BDNF levels were much more evident in transgenic mice treated with pilocarpine. And here the quantification. So all these experiments were gain of function, but we wanted to perform also loss of function experiments. Before that, again in collaboration with the team of Albert Giralt, we perform again electrophysiological recordings in anesthetized mice. But this time the experiment was a bit different. What we did is to retreat the two groups of mice, wild type and GFAP BDNF transgenic mice, with cyanuric acid with a low dose. And 5 minutes later, we induce a train stimulation, and then we record the electrophysiological activity. And as you can see here, not too much things were observed in wild-type conditions, but in transgenic conditions, after train stimulation, the frequency of neuronal firing and the timing, the period was much longer, and the frequency was also very high, much higher than compared to wild-type conditions. Okay, and now yes. So all these experiments were performed as a gain of function experiments, but we also wanted to verify these readouts in these experiments in loss of function experiments. To do so, what we did is to use primary cultures, neuronal primary cultures, and we treated them with 4-aminopyridine, which is the 4AP model. And here you can see the basal conditions and the 4AP conditions. You can see that upon treating these cultures with fluo4, we can record the activity, the calcium-dependent activity of these neurons, which is much more evident in the 4AP treatment than basal conditions. So what we did in this experiment as experimental design is, as I told you at the beginning, more or less, a very similar experimental design, in which we co-cultured wild-type neurons with wild-type astrocytes, and we compared them with wild-type neurons co-cultured with BDNF knockout astrocytes, and we subjected these two conditions to this model of 4AAP. Here you have a recording of this calcium activity in basal conditions, which is this activity, compared to 4AP condition, which is this. Here you have a representative video of this activity. To evaluate and to monitor these results, we used the NetCal software, which was developed by the Jordi Soriano's team, in which we can infer the number of spikes and the global neuronal activity in order to evaluate synchronic-like activities in a primary culture, in a neuronal primary culture. What we observed is, first of all, the firing rate in individual neurons was increased in both conditions. So upon treatment with 4AAP, the firing rate increased per neuron equal in wild-type neurons co-cultured with wild-type astrocytes, and in a similar rate in wild-type neurons co-cultured with BDNF knockout astrocytes. However, the global activity measured in two different parameters, like the active neurons or the global firing rate, which is a measure of the global activity, the increased number of activating neurons. We observed that the number of activated neurons increased in the condition of BDNF, in the condition of wild-type neurons co-cultured with wild-type astrocytes, but there was no increase in the number of activated neurons in the condition of wild-type neurons co-cultured with knockout BDNF astrocytes. And here we have a similar result, but analyzed in a different way. I don't want to disturb you by explaining the different parameters, but in conclusion, what is saying the result is that BDNF from astrocytes can play a key role in the synchronization, in the increased synchronization or the global activity in epileptic-like models. So just to finish this part and the entire talk, it is noteworthy that it's not so simple like this. Our group and other groups have indicated that BDNF, depending on the moment of the disease in the context of temporal lobe epilepsy, can play a positive or a positive role in neuronal survival or a detrimental role in neuronal survival. As here you have a representation of this model in which upon induction of temporal lobe epilepsy, there is an increase, a massive increase or huge increase of BDNF levels. And the BDNF in this moment or in this stage of the disease is aberrant, is detrimental, and plays a role in excitotoxicity. So if we administrate BDNF at this moment, we will exacerbate the phenotype or the readouts, or we will exacerbate the neuronal damage. But in contrast, if a BDNF is administered in a phase in which there are no acute or toxic events, in a standby phase, let's say, this BDNF levels administration could be even though neuroprotective because it's helping or trying to save neurodegenerating neurons. So just as a take-home message, we have provided, I think we have provided some grains of sun in the field of the yin and yang role of BDNF in the context of neurological disorders. And we think that, well, we think that we have demonstrated that increased BDNF delivery from astrocytes induces structural synaptic plasticity and rescues synaptic function and plasticity in Alzheimer's disease and Huntington disease models. But in contrast, increased BDNF delivery from astrocytes can induce excitotoxicity in some conditions, or it exacerbates neuronal death in temporal lobe epilepsy models. And so that's all. And just to by ending this talk, the most important part, the part of the acknowledgements. So the institutional acknowledgements, the funding, and the current people in the team. We are a small team, but super powerful. Former people like Benoit Depens, Sara Fernandez from the Albert's team. The most important collaborators, Jean-Antoine Giraud, Jordi Albert, José María Delgado, Agnès Gruart, and Sylvia Ginés. And I think that's all. Thank you very much for your attention and patience to hear my talk, because maybe I was a little bit fast, but I hope clearly enough. So that's all, and thank you very much.
Well, thank you very much, Albert, for a very nice presentation. So the paper is now open for discussion. If anyone is interested in making a question to Albert, please do so by either raising your hand using the option in your screen, or by sending me a message stating your name and your interest in making a question. I will give the floor to make the question yourself, so there is no need for you to write down the questions in the chat. Okay, so while we wait for any questions, let me just remind everyone that the CIBERNED Webinar Series is held every other week on Thursdays at 3:30 p.m. And you can double check the upcoming webinars in the CIBERNED website, where we have posted a list of webinars till the end of the year. Okay, so any questions? I think there is a question from Mireia Moreno. Mireia, you could make a question yourself if you have a microphone available. Otherwise, I will read the question.
I think I read it. I think she was asking why BDNF levels are reduced in Alzheimer's and Huntington disease. Maybe this is the question.
Yes, that's it.
So there are many potential mechanisms. For example, at transcriptional level in Huntington disease has been shown that BDNF is reduced because several transcription factors have altered their function, like the rest nest dimers and transcription factors. Also, another origin is the transport. So in the Huntington disease context, BDNF levels in the striatum are mostly provided by cortical afferents. And there are many groups that have described, like, for example, Frédéric Sodou in France have described a deficit in the BDNF transport from the cortex. Although BDNF express its own levels of BDNF, they are at very low levels, and striatal neurons depend on the cortical BDNF levels. And since the transport is altered, these BDNF levels are reduced in the striatum. In the context of Alzheimer's disease, it's not so clear. I don't know very much which is the reason of these reduced BDNF levels. I think that there is also a mechanism involving transcription factors too, but I don't remember so good, like in the context of Huntington disease.
All right, thanks, Albert and Mireia, for the questions. Any other question from the audience? You can use either the chat option or also the raise hand option in your screen if you want to make a question to Albert. Okay, there is one raise hand here from Teresa Iglesias. Teresa, go ahead.
Yes. Hello, Albert. Thank you for the talk. I'm curious to know whether you have used this model, your model, the one overexpressing GFP BDNF, in an acute model of brain damage, like a traumatic brain injury or stroke.
No, and this is a very interesting question, because I don't know about this field, if an increase of BDNF is beneficial or detrimental. I don't know if you know it, but no, we never thought about that. The most acute model that we have used is a temporal of epilepsy, in which pilocarpine is an acute model, a toxic acute model. But I didn't explain too much about the experiment, but we evaluated the mice 1 week later or 10 days later, so in which the neurodegeneration and the cell death is super evident. We never focused at the beginning of the pilocarpine induction. But anyway, the question was about traumatic brain injury, and we never tested. And I don't know if it could be better or worse, because, you know, as I told you, there is this bimodal or bidirectional potential effects by an increase of BDNF.
Mm-hmm. Okay, thank you. Thank you very much.
You're welcome.
Okay, thanks, Teresa. Any other questions? Yeah, we have a raised hand from Ian or Ian. Please go ahead.
Hello, can you hear me?
Yeah.
Yes.
So I'm not a neuroscientist, so I'll get my excuses in at the beginning. But if we go with the idea that BDNF is something beneficial and that this has been known for a long time, why is it that nobody's done a screen for compounds that induce BDNF and trialed it in Alzheimer's and other conditions that you've highlighted?
So why is not used in the companies to induce?
Yeah, why is it being exploited?
Well, actually, there are many drugs that induce BDNF expression, and now it's coming to my mind the antidepressants. For example, I don't know if sertraline or some antidepressants induce a huge amount of BDNF increase. Actually, there is a recent work from a team in the north of, I don't remember, in which they demonstrated that antidepressants, all of them, act through the BDNF TrkB system. So, and you know that antidepressants exert beneficial effects in a big subpopulation of the depressive patients. In the case of Huntington disease, there are many drugs that induce a BDNF expression, like HDAC, histone deacetylases inhibitors or whatever, yeah. Or also other, there are many compounds that induce BDNF expression, but this BDNF expression, I don't know, but my idea is that to overexpress or to induce a BDNF expression without control is not so good, because as I told you before, if you express BDNF in tissues that are not suffering for neuropathology, maybe this is not good enough. That's why we designed this tool to overexpress BDNF only when required and where required in the brain regions in which there is disease, and in the brain regions in which there is when astrogliosis takes place. So this is the temporal perspective. And also compounds act through a lot of mechanisms. So some of them are good, some of them are bad and specific. They have a half-life that depending on the chronicity of the treatment, they lose their effects or whatever. So that's why maybe there are another line of research that are looking for the administration of BDNF from gene and cell therapy, or using Trojan horses, or just to administrate BDNF with molecules, nanomolecules that help to BDNF to cross the blood-brain barrier. There are still many caveats and problems to be solved. But just compounds, for me, they are too much unspecific and too much artificial from my point of view.
Yeah, thanks very much.
All right, thank you for the question. I see another question on the chat. Albert, if you can see it, it's also from Mireia.
¿Por qué crees? Well, why do you think that you find out the contrary effect in the temporal lobe epilepsy? Maybe because of the p75 signaling, or maybe because there is more pro-BDNF levels. Well, first of all, not only pro-BDNF levels can exert an excitotoxic function, but also mature BDNF can play a role. First of all is this, and indeed, in the temporal lobe epilepsy, during the seizures, there is an increase of BDNF release. So, and this is toxic because BDNF induces neuronal activity, and you know that when this neuronal activity is too much increased, becomes excitotoxicity. So, if BDNF is implicated in this excitotoxicity, it's detrimental. And the idea, and that's why we observe the opposite effect in the TLE models compared to the degenerative diseases in which BDNF is reduced, because BDNF is already increased, and we are exacerbating this increase, and this exacerbation is inducing even more excitotoxicity. But we cannot rule out the role of pro-BDNF because we also observed an increase of pro-BDNF in wild-type conditions, but the mature BDNF was also increased, and much more in the transgenic mice.
All right, thank you, Albert. Thank you, Mireia, again for the question. Any other questions?
You're welcome, Mireia.
I don't see any raised hands or any question coming through the chat. Let's wait for a few seconds just in case it's a late comer.
Can put a chronometer.
Yeah. All right, so this is the last call for anyone interested in making questions to Albert.
Or alternatively, they can contact me through email if they feel shame or shy or whatever.
Okay.
I'm always open to answer questions by email, so no problem.
Yeah. All right. So I don't see any other message to the chat or any raised hands. So I think it's time to close the session then. Not before thanking Albert for a very nice presentation and for his willingness to share with us his work. And he just said, if anyone is interested in getting into further discussion, please feel free to contact Albert directly through email or phone or whatever. So thank you very much, Albert. Thank you everyone for attending this webinar, and I wish you all a great afternoon. Bye-bye.
Thank you very much for this kind invitation to you too.
Bye.
Bye.